Before we start discussing animal cells, it will be much more meaningful if we observe some animals first. I'm outside the local zoo. Let's go in, check it out. Animal kingdom is made up of diverse group of organisms. Some of them are very simple, others are very complex. First, we are going to look at some simple animals. Sponges are the simplest animals on the planet. No systems, no brains, no hearts. In other words, it's just a colony of cells. Jellyfish are a little more complex than sponges, but still very simple. A few organized systems in the body, no brains, no hearts. They have specialized cells that act as defense cells. The complex animals, they have systems, digestive, blood vascular, nervous system with brain, cardiovascular system with heart, and they are adapted for many, many different environments. All these animals, from simple to complex, their bodies are composed of compartmentalized structures called cells. In this video, we will look at the major parts of a typical animal cell. This is a diagram of animal cell that I have drawn. I've taken out all the extra details. This is a simplified version, and that will help us to understand the animal cell parts. The boundary of the cell is called plasma membrane. The plasma membrane is very, very thin, and it's made up of proteins, carbohydrates, phospholipids, and cholesterol. It has a very complex structure. We need a totally separate video in order to understand this structure. But when it comes to the functions of the plasma membrane, it moves some of the material inside the cell and moves some of the materials outside the cell in order to maintain the homeostasis of the cell. The cell has to be healthy. The cell has to maintain the constant internal environment. And that internal environment is made by moving some of the materials in and some of the materials out. So the plasma membrane plays a major role in selecting the materials to move inside the cell and selecting the materials to go outside the cell. And that's why it's called selectively permeable. And this selectively permeable makes sure that the cell has a good homeostasis. Usually in the center of the cell, there is a ball-like structure called a nucleus. This nucleus is called the control center of the cell. The reason behind that is because this nucleus has the DNA. And the portions of the DNA are the genes. And those genes are capable of making proteins for building the cell body. And those genes are capable of making enzymes. They control the enzymes that control all the cell activity, all the chemical activity inside the cells. So the structure and the chemical activity is controlled by the DNA. That's why this is called the control center of the cell. The boundary of this nucleus is called nuclear membrane, and this nuclear membrane is similar to plasma membrane. And inside this nuclear membrane you have some holes or little pores, and those pores actually are called the nuclear pores, through which the larger molecules like RNA, mRNA, they can go through this, and that's what the nuclear pores are for. This whole ball-like structure is filled with a watery fluid called a nucleoplasm. And this nucleoplasm is a medium where the chemical reactions, they can take place inside the nucleus. The last part inside the nucleus is called the nucleolus. And this nucleolus actually makes the ribosomes in here. And those ribosomes, they go through the nuclear pores outside and they get attached to endoplasmic reticulum. And that's what the main function of the nucleolus is, to make ribosomes. Between the plasma membrane and the nucleus, this space, all this space is filled by a watery fluid. And this watery fluid is called the cytoplasm. And this cytoplasm is mainly water, mostly water. And inside the water you have some salts and electrolytes and sugars, amino acids, those kinds of things are dissolved inside. And that's what the cytoplasm is. If we want to make cytoplasm inside our house or inside our labs, we can make it easily. All we need is water, distilled water, and inside the distilled water you can dissolve some salts and sugars and electrolytes, and that's your cytoplasm. Between the plasma membrane and the nucleus, there are some parts that float around in the cytoplasm in here. And these parts are called organelles. The reason they're called organelles is because they function like little organs. They are not organs, but they function like little organs. They have specialized functions, and due to those specialized functions, they are called organelles. The first organelle inside the cytoplasm is called mitochondrion. Mitochondrion is singular. Mitochondria is plural. And these mitochondria, they are called the powerhouses of the cell. The reason behind this is that these are the places for cellular respiration. The sugar molecules, the carbohydrate molecules, they go inside the mitochondria, and most of the cellular respiration takes place in here. And during the process of cellular respiration, the glucose molecules are converted in energy, and that energy is stored in ATP molecules. And that's what takes place in here in the mitochondria. And that's why they are called the powerhouses of the cell. In our thigh muscles, in our arm muscles, we have a lot more mitochondria. In our cells, we have a lot more mitochondria in those places where we use those muscles to produce a lot more energy. We need a lot more energy in those muscle cells. But if you think about the neck muscles or the neck cells right here, they don't have those many mitochondria, because these cells, they don't need that much energy. They don't produce that much energy. That's why they don't have that much mitochondria. Inside the cell, we also have two other organelles. They are sac-like structures. One is a double membrane bound sac-like structure called the lysosome. Another one is a single membrane bound structure called the vacuole. The lysosomes, they're filled with enzymes. And what they do is they go close to the food molecules and they burst open, they release all their enzymes, and those food molecules, they get digested. And sometimes they're also called suicidal vesicles because they commit suicide. When they commit suicide, and more of these lysosomes are going to be made by the ER after they are disappearing. And that's the lysosomes. They help in digesting the food molecules, in other words. But the vacuoles, when it comes to the vacuoles can be sacs of anything. They can be sacs of food, sacs of water, sacs of waste materials. So those are the vacuoles inside the cell. So the two organelles, sac-like organelles inside the cell, vacuole and lysosomes. Inside the cell, we also have ribosomes, those little dot-like structures. They are made by the nucleolus right here. They come through the nuclear pores. They get attached to the endoplasmic reticulum, and they are called ribosomes. Some ribosomes are free-floating, and there are only few. They're in the cytoplasm. They're free-floating. What is the main function of the ribosomes? The ribosomes, actually, these are the sites for protein synthesis. That's where the protein synthesis takes place. mRNA, all these other associated molecules, they work in here on the ribosome, site of ribosome. They produce the proteins, and those proteins are poured in the endoplasmic reticulum. And that's what the main function of those ribosomes are. They are the sites for protein synthesis. Inside the cell, we have two other sac-like structures. One, these are the tubules and the sacs, the network of tubules and sacs. One network of tubules and sacs, they have ribosomes attached to this, and that network is called the rough endoplasmic reticulum, because under the electron microscope, they give a rough appearance, the beaded appearance. They look like beads are attached to this, and that is the rough endoplasmic reticulum. The smooth endoplasmic reticulum does not have the ribosomes attached to this. That's why this is called a smooth endoplasmic reticulum. So two kinds of endoplasmic reticula inside the cells, and that's how they look like. Physically, structurally, they appear to be different. When it comes to the smooth endoplasmic reticulum, it helps in lipid synthesis. The lipids and fatty acids are made in here, and it helps in carbohydrate metabolism. The carbohydrates can be metabolized. More carbohydrates can be made in here. Sugars can be made in here. And the last part is that detoxify drugs and poisons. If by mistake or by chance the drugs and poisons, they reach inside the cells, the cell has a process in here. In the smooth endoplasmic reticulum, you have some enzymes. Those enzymes, they will work on the drugs and poisons, and they will detoxify them. They will change them into non-poisonous materials, and that's one of the main functions of the smooth endoplasmic reticulum. People who are drug users, when they consume drugs on regular basis, what happens is those drugs, they start going inside these cells. When they start going inside these cells, this much smooth endoplasmic reticulum is not enough to detoxify those drugs. So what happens is that the smooth endoplasmic reticulum will start expanding inside the cells. So if you look at the cells of drug users, their cells, they show a lot more smooth endoplasmic reticulum as compared to a person who's not using drugs. When it comes to the rough endoplasmic reticulum, there is a ribosome. The ribosomes are attached with this. Those ribosomes are a site for protein synthesis. The proteins are synthesized in here. Those proteins are poured inside the rough endoplasmic reticulum. And this rough endoplasmic reticulum is going to take those proteins from one part of the cell to the other. And that's what their main function is. They transport proteins from one part to another. Inside the cell, there's other organelle that looks like pancakes. And this pancake-like structure, these pancake-like structures, they are called Golgi apparatus or Golgi body. This was discovered by an Italian scientist in 1898. He was looking at some nerve cells under the microscope, and he saw this organelle that was not described by anybody, and he named it as Golgi body or Golgi apparatus or Golgi, just the Golgi. The main function of this organelle is it helps in secretion. There are two main kinds of things. One is excretion, where we throw out the useless material or the waste materials. But secretion means when the cell throws out the useful materials. The materials, those are healthy. The materials that are good. The materials that can be used some other place. And that's secretion. Inside the cytoplasm, we also have two structures which are called centrioles or centrioles, and these centrioles, they help in production of microtubules during the cell division. When the cell divides, the chromosomes, they move on the spindle-like structures, and those spindle-like structures are made by the centrioles, and that's what their main function is. They help the cell in the cell division process. The last part is, if you think about the cell, it's a bubble of water. And the plasma membrane is mostly phospholipids. So there is a very high chance that those bubbles of water, they will burst and everything will be scattered all over the place. So there is cytoskeleton. The cells, they have cytoskeleton. They have protein tubes, which are called microtubules. They have protein fibers that are called microfibers. Both of those things, they make a network inside the cell. Sometimes those protein filaments, they go from the plasma membrane to the nuclear membrane. So they anchor all those softer parts inside the cell, and they create a kind of endoskeleton in here. And that's what the cytoskeleton of the cell is, in order to make sure the cell stays safe and the cell does not burst and the cell is healthy. And that's what the main function of those endoskeleton is.